Okay, welcome everyone to our Gateway to Translation webinar series. I'm Sean Knight. I'm an academic respiratory physician working at the Lydia Becker Institute over in Manchester. And it gives me great pleasure to introduce Stig from Exhalation Technologies today. Stig has a master's degree from Copenhagen, and he also has a business degree from the Copenhagen Business School, and has been working in product development, both in the strategy section and in actually developing products for the last 35 years. And it gives me great pleasure to introduce Exhalation Technologies, the company he works for now. So I first met Stig at a Translation Manchester event earlier this year. And Stig really impressed us with his Inflammatory device, which detects inflammatory byproducts in exhaled breath. We've got a project together where we're working on the early detection of lung cancer. And it's really good to see that Stig's going to be talking to us today about lung cancer and how Inflammatory and the technology underpinning it can be applied to this very important clinical question. So with no further ado, I'd like to hand over to Stig now to take us through his very interesting talk, and I'm really looking forward to hearing it.
Thank you, Sean. No need to introduce myself then, but thanks to all of you for tuning in. I'll be talking on biomarkers today and the potential disruption of lung cancer screening that we believe can be achieved by applying biomarkers. And I've been working in this field for more than 10 years, and we keep on hearing we need more biomarkers. And this is actually across a cohort of worldwide diseases and conditions. When we dig into it, we see there's a recent publication saying that there's an increase in likelihood of having a drug approved if you include biomarkers in your development strategy, and it's a significant increase. There are other stating that the existing diagnostic tools are imperfect in terms of diagnosing everything we want to know, and therefore there's a need for additional biomarkers. So it's a broad need that's been defined. When we talk biomarkers, it's essential to do so in the context of what they are to be used for. If we use them in terms of developing new drugs, we typically use them at the early stages. Oh, sorry, not this is to understand diseases. It's to generate and test hypotheses. And once we gain a better understanding, then we can start to identify and validate even more biomarkers, which could potentially then be used in clinicals. In the therapeutic development, we use biomarkers very much in the same way as we do when we use them in clinical decision making. But typically we are using them, say, we can use them before they've been approved for clinical use. And also they can really leverage development. But for today, I'll be focusing on the decision making in clinical practice and how biomarkers can be used there. If you turn to institutions like the National Institutes of Health, they will provide you with different ways of looking at biomarkers and grouping them. And it's really not the grouping that I'm that interested in. In reality, when looking at it, you may realize that a few or quite a lot of biomarkers could potentially fall in different groups or in multiple groups. What's interesting about the grouping is that you need to understand what it is you want to use your biomarker for, because the requirements to the biomarker, as well as the requirements to how you can measure it, depends on the utilization. At Exhalation Technology, we test in breath. Breath is a rich source of biomarkers, and furthermore, which is equally important, it lends itself to non-invasive testing. And if you're clever and if your technology is right, you can even do it at point of care. When you open up breath and look into the compounds of it, there are different groupings. There are gases where we're using pheno or fractionated nitric oxide to test for eosinophilic inflammation in asthma. We're using CO2 both diagnostically and to monitor anesthesia. One of the big areas that's being researched heavily is the VOC, so the volatile organic compounds. I'll get back to that. Most of these volatile organic compounds are found in the gas phase, but some are even found in water vapor, where we also find markers of oxidative stress like hydrogen peroxide and pH and isoprostane. Aerosols, there's a fourth element in the breath, which is particularly interesting, I believe, because that gives us the ability to even detect virus, especially if we have a virus like SARS-CoV-2, which was spread by the breath, actually carried in aerosols. If we can find ways of detecting it, if we can find ways of capturing the aerosols, we can develop tests, diagnostic tests, which are not just based on biomarkers, but which are actually targeting the final target. Also, in the aerosols, we find proteins, surfactants, DNA, RNA material, stuff which is highly relevant also in the lung cancer screening context. And finally, the breath itself has physical characteristics like the flow. So if you are an asthmatic and you have issues in exhaling, this can typically be seen in the flow pattern. Likewise, temperature and relative humidity have been researched and are also biomarkers. Now, as I said, VOC, so volatile organic compounds, that's a huge area. In total, if we look at the biological fluids in the body, more than 2,700 different VOCs have been identified, and that's in healthy individuals. More than half of that, almost 1,500, have been found in the breath. The table here is from a study from 2013, where they did a review of existing literature to establish which VOCs could actually characterize which lung diseases. And as you see, it dates back all the way back to 2003, so it's not like it's new. And quite a lot of them are also addressing lung cancer. That was also the topic of this review. So there's a huge portfolio of VOCs that could potentially help as biomarkers in the screening and diagnosis of lung cancer. If we then look at how do we bring a biomarker from being an idea into being something which we can use clinically, well, in reality, it's very simple. We have a new biomarker, we find a way of testing for it, and then, assuming that our ROC curve comes out with an area under curve of more than 90%, then we have a good classifier. However, that's rarely reality. So normally we start below 90%. Sometimes we are even pleased if we find 70 or 75%. But as an outset, that's not a good classifier. So therefore, we start looking at multivariate associations, taking into account risks, odds, hazards, and we also look at the significance clinically as well as statistically. And based on this, and then assessing the value in existing clinical models, it can actually turn out to be very useful despite having a lower area under curve. One thing that's driving development now is the fact that we have become better and better at introducing nanotech-based sensing technologies. In reality, what it means is, of course, you should be selecting a technology which matches the biomarker you're targeting. But by bringing in the nanotech technology also means that we can drive towards point of care. Because one of the challenges in testing for biomarkers have been that we have typically been looking at very low levels of biomarker contents. So detecting it has been an issue. But with the new technology, it's coming up where we can actually do it. And it's not just new technology. The electrochemical biosensors have been used for years, and they actually provide similar opportunities. So we all know of the glucose testing in diabetes, which was done in a drop of blood, so people could monitor and manage their own condition, and that was using a biosensor. So it's been around for a while. One thing that's new and really important in this context is the emergence of AI. And now AI, I don't believe AI is the solution to everything, but a key thing is that AI expands on processing capabilities that we've never had before. So now we're able to actually process huge sets of data. We are able to combine inputs from different sensors to an extent which we've never been able to do before. And that really moves boundaries. Now, if we look at the use case, which is the main theme of today, we're looking into lung cancer. And the burden, I guess, is known to all of you. The fact that it's diagnosed too late in general, meaning that only 4 in 10 people will survive, on an average, will survive the first year after detection. The fact that it's actually the highest load in terms of health costs in Europe. And it's also a huge load on patients and families. And if we look at the way we've been diagnosing lung cancer, so traditionally, it's when you diagnose it based on symptoms, then it's basically too late. So there's, if you look across a timeline, there's from the time when the first cancer cell emerges or introduces itself until you can actually see it on a CT scan, there's a period. But then once you can see it on a CT scan, then typically people have been CT scanned for other reasons. And then incidentally, we've detected cancer. And that also means that the early diagnosis, which is a prerequisite to curative treatment, that only applied to 25% of the patients. Now, in the UK, we've introduced the Targeted Lung Health Check program in order to address the need for more efficient lung cancer screening. And we start by looking at people between the age of 55 and 75, which is in total around 15 million. And we do a risk assessment, primarily looking at their smoking history, as we've realized that smoking is a primary driver in lung cancer. So approximately 13 million of these 15 million are ineligible for screening by the current threshold. So they may have smoked, but they haven't smoked too much, and thus there's no further action planned. The remaining 2 million will be eligible for screening. In the UK, we offer low-dose CT scanning every second year. Most other countries I've been looking at offer annual screening, but that's a decision to be made. If the low-dose CT scan shows an anomaly or something that doesn't look right, we move into further investigation. It could be more screening or scans, but it could also be a tissue biopsy. And if the tissue biopsy comes out positive, then we move into classification of the tumor and treatment. Now, luckily, to those people undergoing screening, quite a large number of them come out negative. So from the low-dose CT scanning, it's say in the range of 90% that are negative. However, there's a false positive rate, which is fairly high. So another 8, sometimes 9% point are actually found to be negative during further investigations, and no further action is required. If we look at it from a performance perspective, the detection rate in the UK, when we announced the Targeted Lung Health Check program and that it would be rolled out nationwide, it was stated that we aim at screening around 1 million people every year. And we expect to find a little less than 10,000 new lung cancer cases by doing so, which converts into a detection rate of 1%, meaning that 99% of those being screened come out negative. At the same time, we also see from different studies that more than 35% of those developing lung cancer are actually, by current criteria, ineligible for screening. So they're being missed out. In addition, you can make the observation that we expect to find 10,000 by screening, and we expect to detect around 30,000 every year. So there are 20,000 basically unaccounted for. So they're not being caught by the system the way it's set up now. If we then look at the opportunity, if we want to disrupt this patient journey, we identified two areas or two positions we'd like to take. So the first one is we would actually like to improve the risk assessment. If we could qualify people better for the low-dose CT scanning, so we don't scan all those people who don't need scanning, that would be the first take. Secondly, but this will require a lot more evidence, if a screening is adequately strong, then in reality, we wouldn't need the low-dose CT scanning for lung cancer screening. So it could be, at least to some extent, replaced. These are the opportunities we're looking at. And lung cancer screening, why is it interesting to look at it? It comes with a set of pain points. First of all, the CT scanners, they are a bottleneck in themselves, in the sense we don't have an unlimited amount of CT scanners. And also, we're short of radiologists. I saw a study stating that we're missing in the range of 3,500 radiologists currently in order to do what we want to do. Further, doing low-dose CT scanning is expensive, so there's a cost issue in it. Of course, it's also an issue that we miss out quite a big chunk of the new lung cancer cases. Whether it's a waste of limited resources or whether you just call it an inefficient utilization of limited resources, but having a detection rate of 1%, it's of course good for the 99% who test negative, but would be even better if they didn't have to undergo low-dose CT scanning. Likewise, the whole high false positive rate, which comes with a good detection rate, also means that quite a lot of anxiety is being caused, as well as unnecessary costs. On top of this, and this is a different type of issue, we have a fairly low acceptance rate in those countries that are running lung cancer screening programs today. And looking into that, now I guess Japan is the odd one out in this context. They have been doing lung cancer screening for 20 years or so. But if we compare ourselves to the US, they plan to test in the range of 15 million or 14 million people a year. And the adoption rate, so the number of eligible people actually making use of the lung cancer screening, is only 5.8%. So a question is, of course, why is that? And a set of main factors impacting the adoption have been identified. So the first one is political prioritization of lung cancer. In the EU, as well as in the UK, so in Europe, lung cancer has been recommended for a while, for quite a while, for a few years. But the actual decision-making process, because it's a huge investment, takes quite a long time. And some countries even bail out or only do it half-heartedly. Secondly, the way it's set up, where you typically have to do centralized screening, comes at a cost to the people who are to be tested. And that may be part of the reason why they don't show up. There may be issues in simply supporting it from an infrastructure point of view. And finally, there are the more intangible soft issues, like awareness of what is lung cancer, what are the risk factors, and what can screening do, and then even cultural views and stigma related to lung cancer. If we, by identifying the right biomarkers, can come up with a low-cost solution which can be used at point of care, we could, in reality, move the lung cancer screening efforts into the GP's office or provided at a pharmacy. So for the people who are to undergo screening, they won't have to go to a special place. They will go where they're going anyway. From a political point of view, this would reduce cost, making it much more attractive. And policies, at the end of the day, relate very strongly to cost and benefit. So it might have an upside also in the political prioritization. And being able to do it at the GP's office will also improve infrastructure issues dramatically. So that's really what we're driving for. Now, our working hypothesis, and I'd say this is highly down to Sean. He's been doing the thinking. I'm only bringing the message. But the observation is that in the ideal world, we would be targeting biomarkers based on direct tumor products. Whenever you have a tumor, byproducts will occur, typically being seen in the blood. The issue we are having is that when it starts, the signal is extremely low. We may be able to detect it, but it typically requires extremely expensive equipment, which we also don't have unlimited amounts of. At a later stage, the burden increases, and it increases with the cancer burden. But the issue is that as it is at a later stage, it may actually be too late. So the working hypothesis is that what if we target inflammation-based biomarkers or markers of oxidative stress? So as soon as you have a cancer cell present in the system, many immune cells can interact with it, and it can actually cause different reactions, leading to different products being emitted, and it could potentially even be detected in the breath. So this is really the working hypothesis. We believe we are able to detect lung cancer at early stages by looking at inflammation or oxidative stress biomarkers. Now is probably the time to talk a little about why I'm here today. And I believe our background as a company is slightly different. This may be new to some of you, but racehorses have major respiratory issues. Approximately 50% of all young racehorses suffer from respiratory conditions, to the extent that it actually impacts their ability to compete. Now, our founder and CEO, Helle, has a big heart for horses, and she was working in the pharmaceutical industry during the 90s. She was selling products for asthma treatment and allergy treatment to primary and secondary care. And she made the observation that for horses, the only treatment that was offered was systemic treatment, which was inefficient and hard on the horse. And she didn't understand why they didn't have something similar to the baby masks that we use jointly with puffers to deliver medication. So she developed a device specifically for that and brought it to market globally. And she's even selling it today, almost 25 years later. And she's even sold it to Australia to enable medication of dolphins. So it's been around. In the process of developing this, Helle wanted to test the efficacy of her solution. She wanted to verify that she got the kind of spread in the airway system that she wanted to have. This was because of her pharmaceutical background. She knew that was a prerequisite. And when she showed up in Newmarket at the Animals Health Trust to have it tested, one of the researchers approached her and said, "Whoa, Helle, now you developed a solution so we can deliver medication. Wouldn't it be nice if we could also diagnose?" And the challenge when diagnosing horses is you cannot do an interview. And the only diagnostic tests available are highly invasive. They require sedation of the horse, and there's a true risk that you may actually damage the tissue when you do the test. So this guy had developed a system which, well, most of it resembled a horse connected to an industrial vacuum cleaner, but it was really about testing in biomarkers. So Helda said, okay, let's do this. And then what did she want? She wanted a biomarker which was fairly independent from the breathing pattern. You cannot explain a horse how to breathe, or you can explain it, but it will not obey. Furthermore, she wanted it to be available for everybody working in stables. So it shouldn't be a specialist tool. It should be something that you could use to monitor your racing horses. And this is really the starting point, which has taken us to where we are today. So by 2014, we decided to expand on the technology we had and develop a solution which was suited for usage in human beings. Our target when we test is hydrogen peroxide in exhaled breath condensate. Hydrogen peroxide is a very well documented biomarker in respiratory conditions. It's a broad marker of oxidative stress. But if we then zoom in on lung cancer, it also has the potential to become a feasible biomarker in lung cancer. There are not that many studies on it, but the studies that, and that's primarily, I guess, because it's been difficult to measure. But the studies that we've found agree that if you have lung cancer, it leads to an excessive level of hydrogen peroxide. And furthermore, they also observe that this appears to happen as soon, say even at early stages, as soon as you have lung cancer cells present. So this is something which is really interesting. Now, when we CE marked our product in 2019 and presented it in Madrid at the ERS Congress. The same winter at the British Thoracic Society winter session, we presented the results from a small pilot, cross-sectional pilot we'd done, looking across different conditions, including, of course, healthy and lung cancer. And if you look at it, the outcome that we had, these cancer patients, it's 20 as far as I recall in total, but they were at different stages. So it's not just late stage, it was stage 1, 2, 3, 4, so all stages. And there's a very strong correlation with the presence of lung cancer. So the sensitivity appears to be there. However, if you then open up the same box plot and include the other diseases, it becomes evident that the specificity is challenged. It can be difficult to distinguish between lung cancer and other conditions. Furthermore, with collecting exhaled breath condensate, as well as if you collect breath in general, a number of things have been identified that may potentially impact your sample, and you need to take all these into account, or at least you need to be able to argue why it doesn't matter, or why you manage to actually control it in a way so it doesn't have any impact. But these need to be borne in mind. Furthermore, if you look at the typical way we've been doing testing in exhaled breath condensate, we've had a separate device for sample collection. Some of you may know of the R-Tube or the TurboDeck or the Medivac systems. Quite a few research groups have developed own systems because they had other ideas as to how they wanted to collect. So the sample collection has been done in many different ways under many different conditions. Storage and transportation is typically done at -80 degrees. But there's no guidelines on how long, for how long time you can store something like this. And analysis is then taking place in different systems as well. It could be photospectrometry, mass spectrometry, RT-PCR systems, you name it. When you have that much variation in the way you do it, it eventually leads to variation in the outcome. So if you look at literature on hydrogen peroxide, for instance, typically what they conclude is that the H2O2 level in exhaled breath condensate correlates strongly with the level of oxidative stress. However, they cannot agree on a cutoff point because the cutoff point apparently changes across different ways of doing the testing. And this was something that we wanted to overcome. So what we did when we started was we wanted to integrate all of it. And that's what Inflammatory is about. So when you do a test with our system, you breathe into a small handheld device, and condensate is being built up in a condensation area, and a film is building up. It stays there until we have enough, which is typically not a lot. So normally you'd be breathing for 1.5 minutes, 2 minutes, and then the sample releases and flows into a sensor capillary. So we have an instantaneous wetting of the sensor itself. The sensor is an electrochemical biosensor, which is coated with different enzymes, including horseradish. And once the sample is on the sensor surface and a small dot of salt is dissolved in the sample, an enzymatic process starts taking place. We let it rest for a grace period of 50 seconds to let it settle, and then we do our tests. When we test for hydrogen peroxide, it's a fairly simple test because the enzymatic reaction that produces a nanocurrent, which we're then locking. And we do the conversion from nanocurrent over 20 seconds into a final test result simply by calculating area under curve. So it's fairly simple and robust. In addition, and this was driven by ATS and the ERS, we're also locking different other parameters during the sample collection. That was the recommendations made in order to address some of the differences in the way you collect. So if we can describe the collection, the actual collection time better, then we can compensate. So we lock CO2 content in the breath, the flow, the relative humidity, and the temperature, and we do it 10 times per second. So if you're breathing for 2 minutes, you will have 1,200 data points for each of these, and that produces a rich breath print, which we then use, and we combine it with our hydrogen peroxide signal in order to see if we can improve the specificity and even add to the sensitivity of the system. Now, I cannot reveal all of the details on this because it's something which is being published in the very near future, but say the conclusion is that if we use, say, the proper statistical advanced separability algorithms, we can effectively differentiate between various diseases. So when we look at the outcome across 7 conditions and healthy controls, we're able to detect lung cancer and discriminate lung cancer from all the other conditions as well as from healthy controls at a performance level of around, say, 87% area under the curve. And this is early days data. We expect it to improve significantly as we build a bigger base of data. Now, Sean mentioned that we are doing a study jointly, Sean's team and us at the University of Manchester. We were lucky last summer to be granted a translational award by the Wellcome Trust. And what we use it for is to do a pilot study where we're looking at early stage lung cancer. And we're looking at subjects being eligible for screening under the targeted lung health check program, as well as people not being eligible, and likewise for the non-cancer controls. And then we have 2 legs. So one leg is using the Inflammatory as is, starting by looking at the hydrogen peroxide, but also taking the other parameters into account. And then the other leg is using the same device, but simply for collection of breath condensate, And then we do analysis, a lab analysis into the contents, so lipids, proteins, what have you, to see if we can find additional markers that we can use to amplify the performance of the system. So if we can detect the best marker combination for diagnosis, that's really the ultimate objective. Now, this is a fairly small pilot. We're running health economics analysis in parallel, but we want to use this as the foundation for future funding, where we will fund a bigger study aiming at two things. First, establish the amount of data required to make sure that the model we have in our system is adequately robust in terms of translating data into results. And secondly, also looking into clinical applications, so supporting a clinical adaptation. That's what we are aiming for. Now, to sum it up, lessons learned. Now, our company is a small startup, and we've primarily been funded by friends and family. I was shocked, I have to admit, when I read that the NHS themselves assess that it'll take 17 years for a device to be adopted. In 17 years, I'm going to be 80 years old. And if it is 17 years, I'm afraid I will have lost all the friends who are currently funding us. From a small private company perspective, 17 years is simply not acceptable. Furthermore, if you read into the technology strategy, it lacks the solutions that could provide faster adoption routes. And they do exist. We see it in Germany, we see it in France. We need to do the same thing in the UK. So because otherwise, devices will not come to market, because in the private sector, 17 years is simply too long. And also, if it's not to be privately funded, then we need to secure the public funding, and that's also not really being addressed. So that was one thing that was concerning. Another thing is, I think all of us who develop new technologies, we have this dream of coming up with a disruptive technology. We really want to change something, just like we want to change lung cancer screening. By the time when Corona hit us, we had just launched our Inflammatory device, and we were working with lung clinics across the UK, and they were effectively shut down just 3-4 months after we had introduced ourselves. This meant that we could either hibernate, or we could try to see if we could make something up from the new situation. And we decided then that we wanted to develop a SARS-CoV-2 specific sensor. So instead of detecting a biomarker, which is what we do today, we developed a sensor which would pick up the virus. So if you had virus in your breath, it would be picked up by the sensor. And we actually managed to do so. And we managed to, starting from scratch until we had it embedded in a device which we had to change slightly, primarily because we wanted this time, instead of focusing on the breath condensate, we wanted to pick up the aerosols, while at the same time making sure we didn't pick up saliva. So we had to do some changes in the front end of the device to secure that. And furthermore, we had to change it to ensure there was no risk whatsoever of having cross-infections. But we managed to do so in just 6-7 months, which we're proud of. And we then moved into clinical studies in human test subjects. And by December, we had extremely good results. So we ended up having tested in more than 300 people across 6 different sites. We had a sensitivity of 98%, a specificity of 97%. But by December, we only had like 30 positive and 30 negative. But nevertheless, we approached the FDA and we filed an EUA application, so emergency use authorization, by the 31st of December 20. And normally the turnaround time on an EUA application, they promise you it should be in the range of 2 to 3 weeks. I mean, we are talking emergency. However, they just came back with additional questions. And we, of course, invested quite heavily in responding to these different questions. And eventually, by October 22, so almost 2 years later, we got our feedback, and that was that it hadn't been granted and the window was closed. Now, we were a little annoyed about this outcome, so we managed to find a way in and speak to some of the people who had been involved in the process within the FDA. And what they explained to us was that, well, our technology was so new, they didn't know which guidelines to grant an authorization based on. So therefore, they had reached out for their legal counselors and asked them to try to put together new guidelines. And eventually, they didn't make it. So we lost it simply because they didn't know how to approve our device. Now, I'm not saying this because I want to scare people away from the FDA. We actually have a very good relationship with the FDA, and they are being very forthcoming. These days, they appear to be easier to work with than some of the European counterparts. But it's really about having a disruptive technology. What you need to consider is that you may be assessed by standards to which your disruptive technology does not adhere. And that can be a challenge. It can be overcome, of course it can. But we took a hard learning on that. Now, if we go back to the actual journey of biomarker validation, this is how the NIH are presenting it. So basically, you go through discovery and development, analytical validation, clinical validation, through to qualification. And you just have to answer a set of questions, and you should be answering them by yes, and then you're there. So a lot of hard work, but nothing but hard work. However, the Imperial College have noted that according to their data, less than 2% of biomarkers that are being proposed in the science achieve clinical adoption, whereas most of all new biomarkers are stalled or fail, normally after only one paper. I mean, those who actually pay the big price here, apart from them, those of us who find the new biomarkers, is actually the healthcare systems and the patients. Imperial College concludes that they want to qualify biomarkers better, but I think there's another part to this, and that's about valleys of death in this translational journey, which actually starts with fundamental research and ends up in clinical practice and health decision making. And we've identified at least two valleys. So the first valley is the one I think that Imperial College are addressing. And the issue that they're seeing is that if you fail in your first paper, if you don't meet the targets you hope to meet, it's very difficult to keep up the interest and the support you need to carry on. But I'm convinced that for a lot of biomarkers, it may not be the biomarker that's wrong. It may be the way we actually looked at the biomarker. And it may be a feasible biomarker, but it's being stalled at that stage. But once you pass that valley of death, and you move into product development and legislation, it's great fun. I have a background in engineering and R&D. I love that stage. But then you come to the next valley of death. And actually, I have a feeling that it may be even deeper and wider. And that's about the commercialization. So translating a company from being an R&D company into becoming a commercial company. And now I'm talking about startups, because once you're in the bigger companies, they will, of course, have both things. But we as a startup, we need to transition and translate ourselves. And you sit with the feeling that you have your product. It's a late prototype and it's ready. It just needs the final clinical evidence. And then you need to do the registration, and then you need to do the reimbursement, and then you're good to go. When you talk to people, they're all very excited, but in reality, it's very difficult to make people chip in. And I think this is down to risk aversity. So, because once you've crossed that bridge, once you move into growth, then a lot of people say, well, we're here to help you. We want to invest in growth. But taking on the final bit of risk, which relates to each of these valleys, that's very difficult. And at the end of the day, both of these valleys mean that we don't get the technology out there that we want to have. Now, I'm not saying this to tell people that, whoa, you shouldn't do this, it's too much work. I'm saying this because we need to, if we want to bring new biomarkers to market, we need to think ahead. We need to plan with the end in mind. And when I'm in the R&D stage, end in mind is not final product. End in mind is product on market. And I need to engage the stakeholders, the key stakeholders from the next stage, even when I'm in the present stage. And that's, I think, the best answer to how you can cross these different valleys. Having said that, it's thank you for listening from my end.
Great. Thank you very much, Stig.